So what I will present to you today is the clinical good manufacturing methodology and clinical trial preparations for cell therapy using autologous iPSC for Parkinson's disease. Now, Parkinson's disease, as you all know, is a huge problem worldwide. It affects over 6 million people and is actually the second most common neurological disease after Alzheimer's disease. The incidence is increasing because of the epidemiology and the aging and the good aging population worldwide. The problem is well understood in terms of the movement disorder, the lack of movement, akinesia and tremor and rigidity. And it's because in your brain on both sides you have about 1 million dopamine neurons when you're born, but with age, and particularly with age and certain genetic combinations, we all develop a loss of that. And eventually we will need some kind of therapy. Now, this graph summarizes the problem. When you start with 1 million cells, over time, over decades, you start losing that. And when it's accelerated, you develop Parkinson's disease when you lost about 70% of those cells or their synapses, which produce this dopamine transmitter. Now, with Parkinson's disease currently, we've had a treatment for over 40, 45 years of L-DOPA, or dopamine agonist, or a combination of both, even with enzymatic control of the transmitter. However, that standard of care only leaves you with about 5 years before certain things occur. It becomes less optimal and loses efficacy. To solve this, we've worked for decades to actually replace the neuron in cell therapy. We've gone through a number of iterations, which means it's truly become an evolved platform. We started with xenografting here in Boston. We went to stem cell, generalized stem cells, and then we found that autologous cells in monkeys and presumably in humans are really the way to go. And for that reason, we've spent a lot of time trying to perfect this. So what is the healthcare market? Well, it's obviously very big with such a large number of patients, but as I just told you, between 5 to 15 years after the treatment with L-DOPA or agonist, motor complications occur, such as extra movements on treatment or lack of efficacy. And so that is currently treated by either some kind of reduction of therapy, which of course has its obvious effects, but deep brain stimulation actually is a way to work around that, but it's putting electrodes downstream of the lost cell to mitigate and reduce the side effects of the drug. We believe instead that replacing the patient's own cells will be an efficacious way to actually reverse the syndrome and be curative on the movement disorder. So initially, the projection is about 10% of the market, which is a very big market, as I mentioned, in severe patients. But if this works in that patient group, there is no reason in the future to not let patients who have just had the diagnosis get this treatment. And therefore, if this is streamlined, as we heard, we believe a fairly large percent of these patients would like to have a cell therapy. So with data that I will show you in a minute, we are therefore focused on creating a way to generate these cells from the patient. And here is a drawing of a woman with Parkinson's disease who gets her blood drawn. We're currently doing this at Brigham and Women's Hospital. The peripheral blood monocytes are isolated, and then you expand the T cell fraction in the blood, because we have a way, as we just heard, of dealing with T cells. They're reprogrammed with the Yamanaka methods, which is a Sendai virus delivering genes that reprogram your DNA to its epigenetic naked basic form, which means that every cell produced is capable of forming any cell in the body. So it's a platform technology. Now, once you have done that and you freeze them down, there's still important steps that are the new art of cell therapy, namely the QC of iPSC lines. The very hard work, we're working very closely with the FDA, which again creates a partnership where we go through multiple tests for sterility, obviously, but also oncopanels, various ways to determine that these are really safe cells, and karyotyping and so forth. And these are living cells. So once they are expanded and cleared, we can freeze them down again. The patient has a chronic disease, and obviously we have time to develop the cells very well. Our next step is to thaw those iPSC lines, and then with the patented methods, there's one specific patent and 2 patents that we have licensed to a company, we can make the very specific dopamine neurons that die in disease. It may seem trivial, but actually you have several dopamine neurons in your brain, and even some in the retina. So the actual recipe for developing these exact cells has been a challenge, but we have accomplished it. At this time, we freeze down the cells, again do quality control steps, and now have them ready for the patients. So why did we develop this very specific method? Well, we had many years of work leading up to this. As I said, we tested xenografting, stem cell allografting, and finally, we used autologous transplantation. And you see here an example of our first publication in the left-hand panel. A monkey had Parkinsonism, very similar to human, in fact, for 5 years. That then received their own dopamine neurons transplanted in the brain. And these are cells that I worked on for decades. They're dopaminergic pacemakers. They release dopamine very beautifully from the synapse, and if it's too much, they take down the dopamine to normal levels. But as you can see, this monkey develops exactly according to the dopaminergic transmission that occurs with these new synapses. And this is a stable thing, which we've also seen in some people previously with fetal cell transplant, but they had immune problems to some extent. What made neurologists very excited was the data on the right. When the monkeys were transplanted on one side of the brain, which controls the opposite side of movements, the monkeys were able to initiate the complex movement of retrieving actually Fruit Loops under a photocell beam, and clearly from 3.5 seconds could go down to 2, and in some cases in a second series of monkeys, they could come even closer to normal at 1 second. But most importantly, this only occurred when we had successful transplantation. And if you can look in the upper right-hand corner, this is the monkey's own dopamine neurons growing elegantly and effectively into its own striatum, providing new axons and synapses. So at this point, we had a pre-IND meeting with the FDA, and CBER, the branch of the FDA, is very collaborative as they work with cell and gene therapy, and we work closely with them for years to develop an IND-enabling study. And I can tell you here is the results from the in vivo part, which is showing efficacy. So this is using our method, and in the upper right-hand corner you show this is a unilateral Parkinsonism in a rodent. They turn in circles, which correlates very exactly with their loss of dopamine neurons. And in the lower 4 panel, which is part of our IND filing, you can see that minimally effective or maximally effective dose, that is, you can't do 10x like you do in a drug IND, but you can do a low and a high dose to show the range, otherwise the cells would overgrow. But in all cases, in males and female rats, this created a response that is correlated with success. So therefore, in summary, we have ongoing work that will lead us to manufacturing. And we are fortunate to have an NIH grant, the only one of its kind, to actually study this at MGB and Brigham and McLean Hospital. We've manufactured the test article for GLP. We have validated the release assays I mentioned. We have GLP safety study completed. We've done a non-human primate study again to validate our methods. And we have an ongoing GMP manufacturing test at Dana-Farber Cancer Institute using the manual method that we developed in-house. So what's happening next is the IND filing when the histopathological data is completed. And importantly, we will then start an interventional clinical trial funded by NIH money. But we have formed a company because we've realized that not only have we accidentally developed a platform for other therapies in the future, but also this very specific kind for Parkinson's disease, that if we're going to do it in more than 6 patients, it requires what we call closed-loop manufacturing. And we're well underway with our team and with engineers to develop that. And so if we're successful with that, we will be able to go to more patients and do multi-center clinical trials. So the company we have formed, I'm happy to speak with you individually about that, is looking for seed investment for the cGMP manufacturing process to make that complete. Thank you.